Chapter 4 Bacterial Classification, Structure and Replication. Outlines. Objectives: Introduction, Eukaryotic and Prokaryotic cells, Structure of bacterial cells, Classification of bacteria, Nutrition and growth of bacteria, Cultivation of bacteria, Antimicrobial chemotherapy. Objectives. At the end of this chapter, you have to know the difference between eukaryotic and prokaryotic cell, explain the structure of bacterial cell, now the method of bacterial classification, nutrition and growth of bacteria, how to cultivate bacteria, antimicrobial therapy and susceptibility testing. Eukaryotic and prokaryotic cells. Eukaryotic cell. Eu means through. Karyotic means nucleus. The eukaryotic cells has a true membrane bound nucleus usually containing multiple chromosomes. A meiotic apparatus, a well-defined endoplasmic reticulum and mitochondria. Eukaryotes are algae, protozoa, fungi. Prokaryotic cell. Pro means primitive. Karyote means nucleus. The prokaryotic cell possesses naked DNA without associated basic proteins. Divides by binary fission. Example of prokaryotes are bacteria, below green algae. Bacterial cell. General property. Typical prokaryotic cell contain both DNA and RNA. Must grow in artificial media. Replicate by binary fission. Almost all contain rigid cell wall, sensitive to antimicrobial agent. Structure of bacteria. Bacterial structure is considered at three levels. 1. Cell envelope, cell wall and cell membrane. 2. Cellular element, internal the cell envelope. These are mesosomes, ribosomes, chromosomes, nucleoid and cytoplasmic granules, spores. 3. Cellular element external to the cell envelope, flagellum, pilus, and glycocalyx. 1. Cell envelope, external covering outside the cytoplasm, composed of two basic layers, cell wall and cell membrane, maintains cell integrity. A. Cell wall, multi-layered structure and constitutes about 20% of the bacterial dry weight. Average thickness is 0.15 up to 0.5 micrometers. Main structural components are peptidoglycan is repeating disaccharides units and polypeptides. Function of cell wall determines cell shape, prevent lysis or bursting or collapsing due to changing osmotic pressures. Site of action of antibody. Contains toxic components to host. Provides staining characteristics to the bacterium. Based on their cell wall structure, two different groups of bacteria are demonstrated by gram stain, namely gram positive and gram negative. Gram positive cell wall, thick homogeneous sheets of peptidoglycan. Function in cell wall maintenance and enlargement during cell division. Stimulate a specific immune response. Have teichoic acid, lipoteichoic acid. Gram-negative cell wall, composed of an outer membrane and a thin peptidoglycan layer, and also has periplasmic space. Outer membrane is almost layer containing lipopolysaccharides, LPS, lipoproteins, and phospholipids. Is an important factor in evading phagocytosis and actions of complements. Provides barrier to certain antibiotics. Example, penicillin. Digestive enzymes. Example, lysozyme. Detergents. Heavy metals, bile, salt, and certain dyes. Contains foreign proteins in upper layer. Regulate molecules entering and leaving cell. Lipopolysaccharides is one component of outer membrane, has three components. One, lipid A, two, a core polysaccharide, and three, an O polysaccharide. A lipid portion of the LPS embedded in the top layer of the outer membrane, function as an endotoxin. B. Core polysaccharides. Its role is structural. Example, to provide stability. C. Zero or O polysaccharides. Extends outward from the core polysaccharides. Functions as an antigen and is useful for distinguishing species of gram-negative bacteria. For example, the foodborne pathogen Escherichia coli 0157 H7. Note: gram-negative bacteria do not contain teichoic acid, and gram-positive do not contain outer membrane. Components of cell wall of gram-negative bacteria: peptidoglycan, periplasmic space, lipoprotein, phospholipid, lipopolysaccharides are outer membranes. Components of cell wall of gram-positive bacteria: peptidoglycan, teichoic acid. Cell membrane, cytoplasmic membrane. It accounts for 30% of the dry weight of bacterial cell. Phospholipid, bilayer with embedded proteins. It is composed of 60% protein, 20 to 30% lipid, and 10 to 20% carbohydrate. Functions in regulates the transport of nutrients and waste products into and out of the cell. Cellular element internal to the cell envelope. A. Mesosomes, convoluted invagination of cytoplasmic membrane, often at site of septum formation. It is involved in DNA segregation during cell division. B. Ribosomes, cytoplasmic particles which are the site of protein synthesis. It is composed of RNA 70% and protein 30%. Inclusion bodies represents accumulated food reserve. D. Chromosomes, single, circular, double-stranded DNA molecule that contains all the genetic information required by a cell. DNA is tightly coiled around a protein aggregated in a dense area called the nuclear region of bacteria. Plasmid. Bacteria may have extra chromosomal genetic material named as plasmid, small circular double-stranded DNA, which may facilitate survival and propagation of the microorganism, provide virulence and drug resistance. E. Spore. Resistant and metabolically inactive, produced by some gram-positive genera, Clostridium bacillus. Such bacteria have a two-phase life cycle. 1. 0. Vegetative cell, metabolically active and growing. 0. Endospore, dormant cell, produced when starved. When exposed to adverse environmental conditions, capable of high resistance and a very long-term survival. Sporulation, formation of endospores. Germination, return to vegetative growth. Resistance of spore is linked to high level of calcium and presence of TP-cholinic acid. Thick coat, longevity verges on immortality, can live million years. Resistant to ordinary cleaning methods and boiling. Pressurized steam at 120 degrees centigrade for 20 to 30 minutes will destroy the spore. Cellular element external to the cell envelope. Glycocollax, capsule and slime layer contain coating of molecules external to the cell wall made of sugars and or proteins. 2 types. 1. Slime layer, loosely organized and attached. 2. Capsule, highly organized, tightly attached. Functions: protect the cell from dehydration and nutrient loss. Inhibit killing by white blood cell by phagocytosis, contributing to pathogenicity. Attachment, formation of biofilms. B. Flagellum. It is the organ of locomotion in bacterial cell. It is composed of protein named as flagellin. The flagellar antigen in motile bacterium is named as HA antigen. Consists of 3 parts. These are filament, hook, and basal body. The basal body and the hook are embedded in the cell surface, while the filament is free on the surface of bacterial cell. Based on flagella arrangement, this bacteria can be atrichous, bacteria with no flagellum; monotrichous, bacteria with a single polar flagellum; lophotrichous, bacteria with bunch of flagella at one pole; amphitrichous, bacteria with flagella at both poles; peritrichous, bacteria with flagella all over the surface. Fimbriae and villi are shorter and thinner than flagella, found on many gram-negative bacteria. Fibria, singular fibria, can occur at the poles of or can be evenly distributed over the entire surface of a bacterium. Important for adherence. Villi or singular villi are usually longer than fibri. Number: only 1 or 2 per cell. Involved in DNA transfer from one to the other. Classification of bacteria. Bacteria classification depends on the following characteristics: morphology and arrangement, staining, cultural characteristics, biochemical reactions, antigenic structure, base composition of bacterial DNA. Morphology and staining of bacteria are the commonly used characteristics to classify bacteria. Morphology of bacteria. When bacteria are visualized under light microscopy, the following morphology are seen: Coccosi, singular coccus, spherical or oval bacteria measuring about 0.5 up to 1.0 micrometer in diameter. They are found in single pairs, chains, or cluster. Micrococcosi, coccosi occurring single. Diplococcosi arranged in pair. Streptococcosi, chain-forming coccosi. Staphylococcosi, bunches of coccosi or grapes of coccosi. Bacilli or singular bacillus, stick-like bacteria with rounded, tapered, square or swollen ends with a size measuring 1 to 10 micrometer in length by 0.3 up to 1 in which spiral-shaped bacteria with regular or irregular distance between twisting, coiled or cork screw-shaped. Example: spirula and spirochetes. Staining of bacteria. Staining is the process of coloring of colorless object using stains or dyes. Use of staining to observe the morphology, size and arrangement of bacteria, to differentiate one group of bacteria from the other group. Types of staining methods: simple staining method, differential staining method, and special staining method. Simple staining method is a type of staining method in which only a single dye is used. Two kinds of simple stains. Positive staining: the bacteria or its parts are stained by the dye. Example: methyl blue stain, crystal violet stain. B. Negative staining: the dye contains the background and the bacteria remain unstained. Example: Indian ink stain. Differential staining method: multiple stains or dyes are used to distinguish different group of bacteria. React differently with different type of bacteria. Two most common are gram stain, acid-fast stain, or Ziley-Neelsen stain. 3. Special stain: A. Capsule staining method. B. Spore staining method. Bacterial nutrition. Bacteria, like all cells, require nutrients for growth. Most important elements required by bacteria can be divided into two main categories. Physical requirements include temperature, pH, and osmotic pressure. Chemical requirements include source of carbon, nitrogen, sulfur, phosphorus, trace element, and organic growth factor. Chemical requirements: the four most important elements of bacteria are carbon, hydrogen, oxygen, and nitrogen. Carbon: organisms require carbon for the synthesis of numerous organic compounds that comprise bacterial cell. Depending on their requirements, bacteria can be classified as autotrophs, free-living or non-parasitic bacteria which use carbon dioxide as carbon source. The energy needed for their metabolism can be obtained from sunlight or autotrophs, and organic compounds by oxidation, chemoautotrophs. Heterotrophs: parasitic bacteria require more complex organic compounds as their source of carbon and energy. Human pathogenic bacteria are heterotrophs. The principal source of carbon is carbohydrates, which are degraded either by oxidation in the presence of oxygen or by fermentation in the absence of oxygen to provide energy in the form of ATP. Hydrogen and oxygen obtained from water, essential for the growth and maintenance of cell. Based on oxygen requirement and tolerance, bacteria are classified as obligate aerobes, require oxygen to live. Obligate anaerobes grow in the absence of oxygen. Exposure to oxygen kills anaerobes. Facultative anaerobes grow in the presence or absence of oxygen. Microaerophils grow best at reduced oxygenation. Nitrogen for protein synthesis and also for DNA and RNA synthesis obtained from organic molecules like protein and inorganic molecules like ammonium salts and nitrates. Not be careful. Main source of nitrogen is ammonia in the form of ammonium salt. Physical requirements: temperature. Temperature tolerance range is the minimum and the maximum temperature at which a microorganism can grow, which is different in different species of bacteria. Optimal growth range of temperature: the temperature at which the bacteria grow best. Based on different optimal growth temperature requires, bacteria are divided into psychrophilic bacteria 0 degrees centigrade up to 20 degrees centigrade, mesophilic bacteria 20 up to 45 degrees centigrade, thermophilic bacteria 50 up to 60 degrees centigrade. Most human pathogens and many of the normal fibra of human bodies have an optimal temperature of 37 degrees centigrade. Therefore, they are mesophilic bacteria. pH is a measure of acidity and alkalinity. Neutrophilic bacteria grow based on near neutral pH value. Most bacteria grow based in narrow pH range near neutrality between pH 6.5 and 7.5. Acidophilic bacteria prefer to grow at low pH value, acidic medium. Alkalinophilic bacteria prefer to grow at high pH value, alkaline medium. Salinity. Salty content of the medium affect bacterial growth. Halophilic bacteria grow best at high salt concentration. Moderate halophils requires 33% salt concentration. Bacterial growth. It is an orderly increase in all the components of an organism. It is an increment in cell number. Generation time. It is the time taken for a bacterial population to double. Time required for a cell to divide. Bacteria divide into two equal daughter cells and double the number. That means binary fission. The generation time varies from the species, the amount of nutrients, the temperature and the pH and the other environmental factor. Bacterial growth phases. The normal bacterial growth curve has 4 phases: lag phase, long phase, stationary phase, this phase. Lag phase, a period of adaptation with active macromolecule synthesis like DNA, RNA, virus, enzyme, and other structural components. It is a preparation time for reproduction, no increase in cell number. Exponential log phase, the period of active multiplication of cell division. Proceed at a logarithm rate and determined by the medium and the condition of the culture. Stationary phase or equilibrium phase, the period when the bacteria have achieved their maximal cell density or yield. There is no further increase in viable bacterial cell number. The growth rate is exactly equal to this rate. A bacterial population may reach stationary growth when one of the following conditions occurs. The required nutrients are exhausted, inhibitory and products are accumulated. Physical conditions do not permit a further increase in population size. Decline phase, the period at which the rate of decrease of bacterial cells exceeds the rate of new cell formation. There is a drastic decline in viable cell. Few organisms may persist for so long a time at this period at the expense of nutrients released from dying microorganisms.